In this module, we are going to revise how cells divide by mitosis. Most eukaryotic cells can divide by the process of mitosis. DNA replicates in the parent cell during interphase, and the cell then divides to produce two new cells, each genetically identical to the parent cell and to each other. Remember that some cells, prokaryotic cells such as bacteria, do not divide by mitosis, but instead they divide by binary fission. Mitosis is the division used for growth and tissue repair and for asexual reproduction. It is also the method by which white blood cells, B cells, divide to produce plasma cells that then produce antibodies during an immune response. It is also the method by which a cell continues to divide to produce a malignant tumour. The whole process of mitosis is continuous and takes about 20 minutes. We give names to particular stages of the process so that we can recognise what is happening when we observe cells that are in the process of mitosis. There are three phases of the cell cycle: interphase, nuclear division, and cell division. During interphase, the cell grows to its full size and synthesizes many proteins. At some stage, it receives a signal to divide again. Before the cell begins to undergo mitosis, all the DNA in the nucleus replicates. Now, the chromatin consists of two copies of the DNA molecule, or two chromatids. Organelles such as mitochondria and chloroplasts also divide, and the DNA within these organelles is replicated. The centriole also replicates just before prophase of mitosis. There are four stages to mitosis: prophase, metaphase, anaphase, and telophase. Remember the order: P, M, A, T, or PMAT. Prophase. In early prophase, the DNA of the chromosomes, the chromatin, begins to condense and coil, and so the chromosomes take up stains and become visible. By late prophase, each is seen to be made up of two chromatids joined at the centromere. By late prophase, the two centrioles have migrated to opposite poles of the cell, and the nuclear membrane has broken up into small vesicles which are not visible with a light microscope. The nucleolus has also disappeared. Protein microtubule threads are produced and are organized into a spindle by the centrioles. Now metaphase. The chromosomes, each consisting of two chromatids, become attached to the equator of this spindle. The centromeres attach them to the spindle. The cell is now in metaphase. Anaphase. The spindle threads contract and pull on the chromosomes attached to them. The centromere splits and the chromatids are pulled apart and pulled to opposite poles of the cell. Because they are pulled at the centromere, they assume the V shape that you can see here. This stage is anaphase. And finally, telophase. When the sets of chromatids have reached the poles, two new nuclear envelopes reform. The rest of the spindle breaks down and the chromatids now uncoil and form chromatin. A nucleolus reforms, one in each new nucleus, and the cytoplasm of the cell constricts, so that the cell is divided into two. This is called cytokinesis, or cell division. Each new cell has a nucleolus, a centriole, and a nucleus with a full set of chromosomes, identical to that of the parent cell and to that of the other. Here's a cell in prophase, here's one in metaphase, one in anaphase, and this one is in telophase. You may be asked about the biological significance of mitosis. Remember that mitosis produces two new daughter cells, each one genetically identical to the parent cell and to each other. So when an organism is growing or damaged tissue is being repaired, then mitosis is a type of nuclear division that is used. You are continuously replacing epithelial cells in your gut and replacing skin cells. The cell division to produce the new cell is mitosis. Some organisms can reproduce asexually. The hydra shown here is in the process of budding. The smaller hydra is genetically identical to the single parent and will eventually break away and become independent. Many plants can reproduce asexually. These strawberry plants put out runners and small plants develop at the ends of them. They receive nourishment from the parent plant until they can exist independently. One strawberry plant can produce many clones in this way. Potatoes produce stem tubers, each of which can sprout and grow into a new plant. Many houseplants are produced by micropropagation, which is a type of cloning. Meristematic tissue from a plant with desirable characteristics can be cultured in a special liquid or on agar with nutrients, minerals, cytokinins and antibiotics to prevent growth of bacteria. Many genetically identical plants can be produced from very small amounts of tissue. Cells of a mammalian embryo are totipotent. They are undifferentiated and each is capable of giving rise to a complete individual. The cells of a very early embryo can be separated and produce a number of genetically identical individuals, a clone. This technique is important in animal breeding. Agriculturally useful animals, such as cows, have a long gestation period and produce small numbers of young. A female with desired characteristics is treated with hormones, so she produces many ova. She's inseminated with sperm from a male with desirable characteristics, and the embryos are washed out of her uterus. These embryos can be divided and grown for a while in tissue culture and then transferred to surrogates. This ensures that the female with the desired characteristics produces many young but without the risk of being pregnant. The transplanted embryos can be sexed and screened for genetic diseases before they're implanted in the surrogates. This technique can also be used to breed rare animals, as cloned embryos can be transplanted into closely related surrogates. There are ethical issues here, as the animals concerned are denied their natural instincts. Don't confuse this type of cloning with cloning by nuclear transfer, the type that produced Dolly the sheep. During nuclear transfer cloning, an egg cell is obtained and its nucleus destroyed. The nucleus from a somatic cell from the donor animal is obtained and implanted in the enucleated egg. The resulting embryo can then be implanted into a surrogate. However, the success rate for this type of cloning is very small.